So two very important terms when we talk about adaptive immunity are antigens and antibodies. So before we get into the details of it, antigens are basically the markers for the foreign pathogens. So antigens are what we would expect to see on things like viruses and bacteria and fungi. And antibodies are what our body makes, our immune system makes, in response to those antigens. And those antibodies will bind to those antigens and mark them for destruction. So antigens are present on the pathogens. Antibodies are what our body makes in response to those antigens to fight them off. So an antigen is really any substance that your body considers a foreign marker to which it will mount an immune response to. So there are antigens on things other than infectious pathogens, but since we're in microbiology, we're going to focus on antigens present on things like bacteria and viruses. So they are considered foreign markers. Our body would see these antigens and recognize that these things are not supposed to be present in the body and that they should be marked to be removed from the body or destroyed. So in response to antigens present on pathogens, our body is going to make these Y-shaped proteins called antibodies. And those antibodies are designed to attach specifically to the antigens on the surface of the bacteria or virus and then mark them for destruction. The binding site is highly specific, and the binding site that the antibody uses is actually called a paratope. So on the antibody we have the paratope, and on the antigen, which would be on the surface of like the bacteria or virus, you would have the epitope. And this reaction is highly specific. It comes together like two puzzle pieces. So this antibody is highly specific to this antigen. So note that they kind of come together like puzzle pieces. So this antibody here is not going to recognize different antigens present on different viruses or different bacteria. It's designed to attack this specific antigen. So possibly this is the antibody that's designed to attack the spikes on the measles virus. This antibody would not recognize spikes on, say, an influenza virus. So I did kind of already allude to this, but it is a highly specific reaction. So for all the different antigens that are present on different types of bacteria and viruses, our body is able to generate different antibodies. So again, if our body makes antibodies to target antigens on the measles virus, they recognize those antigens only. They're not going to recognize antigens present on the influenza virus. So for all the different antigens that there are available on all the different types of foreign pathogens, our body is very immunocompetent in the sense that it is able to make millions and millions of different antibodies to respond to all those different antigens and mark those pathogens as foreign so we can eliminate them from our body and overcome infectious diseases. So two of the big cells that regulate the adaptive or acquired immunity are T cells, which we often call T lymphocytes, and then B cells, which we'll talk about shortly, or B lymphocytes. So we'll start off with T cells. T cells originate in the bone marrow but do mature in the thymus, hence the name T cell. And when activated, a T cell typically is going to give rise to three different types of progeny. So when activated, a T cell can differentiate into either a helper T cell, a cytotoxic T cell, or a regulatory T cell. Now in our class, we're going to be focusing more on helper T cells and cytotoxic T cells. That's not to say regulatory T cells are not important. However, these cells are more responsible for regulating other T cell responses. So for example, if we have a T cell that goes rogue, and instead of destroying pathogens starts to destroy healthy human cells, the regulatory T cells would essentially kill that cell that has gone rogue. So they kind of monitor the T cells, make sure all the T cells are reacting normally, and if one is not, they can actually induce cell lysis of that cell by a chemical reaction. Because we're in micro, we're going to be focusing more on helper T cells, which help to activate the immune system, and cytotoxic T cells, which are responsible for destroying virus-infected cells. So we're going to start off with helper T cells. So helper T cells do just what their name says. They are responsible for helping to activate the rest of the immune system. So the function of a helper T cell is to activate macrophages, which are phagocytic cells, assist B cell processes, so they can actually assist in activating B cells, which will eventually lead to the production of antibodies. And then they also activate cytotoxic T cells, which are cells that are responsible for destroying virus and cancer-infected cells. So helper T cells are very much important because they do activate the rest of the immune system. That's why HIV is such a scary virus. The HIV virus is known to infect and destroy helper T cells, and by infecting and destroying that one cell type, that virus can essentially wipe out your entire immune system. Because by destroying the helper T cells, it basically prohibits your macrophages from working. So if you get infected with a bacterial infection, phagocytic cells are not going to be available to eliminate the infection. You won't be able to produce antibodies in response to all of your infectious diseases at that point either. And the very cells that would help getting rid of the viral infection, which would be the cytotoxic T cells, they wouldn't be able to get activated because the helper T cell counts are so low. So that virus is pretty fascinating in the sense that it destroys predominantly one cell type, the helper T cells, but by doing that, it can wipe out your entire immune system and make you severely immunocompromised. So with helper T cells, they have specific regions that you need to know. So if we look at this helper T cell here, this helper T cell is going to have something called a TCR, or a T cell receptor. This TCR, or T cell receptor, is what is going to bind to foreign antigen. So the TCR is what is going to bind to the foreign marker that would be present on something like a bacteria or a virus. So it is what actually binds to antigen. Helper T cells also have a co-receptor, and that co-receptor basically just helps the T cell receptor. But helper T cells have a co-receptor called CD4. In fact, you'll sometimes hear helper T cells referred to as CD4 helper T cells. So the helper T cells, they always have this CD4 co-receptor. And with that CD4 co-receptor and the use of their TCR, they are able to interact with other cells that have something called an MHC class II receptor. MHC class II receptors are typically present on cells that we call APCs, or antigen-presenting cells. But antigen-presenting cells are typically going to use their MHC class II receptor to bind to a T cell receptor, helper T cell receptor, and then the CD4 co-receptor is going to kind of help that binding. And once this happens, that will typically fully activate the helper T cell, and then once the helper T cell is fully activated, that's when it's going to release chemicals to go ahead and activate those macrophages, the B cells, and the cytotoxic T cells. So with helper T cells, they are a bit picky in how they work. So they are not just going to go around and actively look for antigen. They want antigen to be presented to them. So they are going to wait for something to present them the antigen, or wait for something to show them antigen. And the cell that is going to show them or present them with antigen, or foreign markers, are the antigen-presenting cells. We have 3 different types of antigen-presenting cells, and those would be B cells, macrophages, and dendritic cells. And what these cells do is they kind of scour through our body looking for foreign antigen. So if we imagine a macrophage just kind of going throughout our body, throughout our tissues, it's going to be looking for foreign pathogen. And when it finds it, it's going to engulf it through phagocytosis, chop it up in a million little bits and pieces, and take a little tiny piece of that foreign pathogen and put it out here on their MHC class II receptor. Because if you remember, antigen-presenting cells, B cells, macrophages, and dendritic cells, they have that MHC class II receptor. So once they've processed that foreign pathogen and taken a piece of that antigen and put it out here on their MHC class II receptor, they are going to go find the helper T cell that's designed to respond to that foreign antigen. But keep in mind, they have to sort through a lot of cells to find the correct cell line. But through this kind of lock and key mechanism, eventually they will be able to bind that antigen in their MHC class II receptor to a helper T cell. That will fully activate the helper T cell, and once the helper T cell is fully activated, it will release chemicals that can then go on and activate additional macrophages to fight off the infection, B cells, which will eventually lead to the production of antibodies to fight off the infection, and potentially even cytotoxic T cells if we're talking about a viral infection. So antigen-presenting cells have a very important job in the sense that they are designed to basically deliver antigen to T cells so that we can activate those helper T cell lines and get the rest of the immune system going to fight off the infection. I've talked about cytotoxic T cells a little bit. Cytotoxic T cells are meant to fight off virus and cancer-infected cells. So with a cytotoxic T cell, they also have a T cell receptor, and that T cell receptor is designed to bind to antigen or foreign markers. But unlike the helper T cell, their co-receptor is CD8. So if you remember, helper T cells have a CD4 co-receptor. Cytotoxic T cells have a CD8 co-receptor, so their co-receptor is different. And using their CD8 co-receptor and the TCR, they are going to bind to cells that have something called an MHC class I receptor. So MHC class I. If you remember, antigen-presenting cells had MHC class II, and they would interact with helper T cells. So what type of cells have MHC class I? The answer, all cells that have a nucleus. So basically everything except a red blood cell is going to have MHC class I. And normally, MHC class I receptor on a cell is empty. There's nothing in it. However, when you become infected with a virus, a small portion of that virus is going to be placed in the MHC class I receptor as foreign antigen. And when a cytotoxic T cell comes along, it's going to recognize that there is antigen in that MHC class I. So the TCR and the CD8 co-receptor are going to bind to the MHC class I receptor on the virus-infected cell, and that is going to cause the cytotoxic T cell to release chemicals that are ultimately going to kill the virus-infected cell. So these cytotoxic T cells, they're highly specific, so they're going to be looking for cells infected with a specific type of virus, but they are going to bind their the CD8 helper T cells are going to bind their TCR and CD8 co-receptor to the antigen present on the MHC class I and release those chemicals to destroy the virus-infected cell. They're not going to harm healthy cells because healthy cells will not have any antigen present on their MHC class I, so that binding will not happen, and those cells will stay alive and normal because they're not infected with the virus. But any cell that's infected with the specific virus that the cytotoxic T cell is designed to fight off, it will be able to bind to and release those chemicals to kill those virus-infected cells with precision.